guys. Welcome to episode 8 of Hairy Matters. Bax and I have the pleasure of being joined by Geoff Hamilton, the co-founder and CEO of Stemson Therapeutics, the leading hair clone company in the world, based out of San Diego. They were founded in 2019, and if their solution is successful, it will be the only solution on the market to actually be able to generate de novo hair follicles, capable of rejuvenating a fully slick bald scalp, which would be a really big innovation for many of the hair loss sufferers out there and many in the hair DAO community. So today we're going to kind of start with the full Stemson journey from basic research, the inception of their company, to where they are today. And then at the end, we'll sort of brainstorm some ways that hair DAO can help support Stemson and work together going forward. You know, there's also been a couple really exciting updates with Stemson recently, so we'll be sure to touch on those as well. So Geoff, first of all, thanks so much for coming on. We really appreciate it.
Yeah, thank you, Andrew, and both Andrews, I should say, really appreciate the opportunity to come on and talk to you and your audience.
So before we kind of dive into the recent exciting updates, both of your sort of xenograph mouse model and the Aderans partnership. Can you provide a little bit of background on Alexei Tursky's, your co-founder's, sort of his basic scientific research, which led to the initial discovery, which then led to the genesis of Stemson?
Yeah, sure. It's a good story, actually, because Alexei is not a scientist who's focused his very esteemed career on hair and hair biology. He actually comes more from the nervous system study and how central nervous system operates from a developmental biology and regenerative medicine perspective. And so he, in about 2010, was looking for an opportunity to do some study where his world of nerve-related cells and his father's world of skin biology — his dad was a fairly esteemed skin biology researcher in Russia — where they could form a joint project. And so they looked at areas where those two worlds of biology intersect. And they determined that the hair follicle is an interesting construct, if you will. It's an interesting model where there isn't a ton of study across the field in academia, which most academics look for white spaces where they can innovate. And at the same time, as they were thinking about this, it was an exciting time in science with the introduction of induced pluripotent stem cells just a couple of years beforehand that had been a strong area of interest for Alexei. And they were wondering, could we join this race to try to differentiate hair follicle related cells from IPSC? Is anyone doing that out there, right? And so they agreed to do a joint study, and they specifically wanted to target the dermal compartment or the dermal papilla compartment of the hair follicle, because so little was known specifically, yet there was a lot of evidence that that was an extraordinarily important and essential component of the follicle. And so they embarked upon a joint project between the two of them, a team in Russia, a team in the Sanford Burnham Institute in San Diego, to try to create a protocol that could differentiate IPSC into dermal papilla. And after a couple of years, they had success. And so it was, again, not, an area that had sort of, I say, strategic focus or intent from Alexei's lab, but he is someone who likes to look at how he takes his skill sets and applies them to tangential areas like this. And they just happened to, have a lot of success, and they published on that and formed patents on that. And then the Institute and him decided, this could potentially have real commercial value. And so for a few years before he and I met, he had been looking at ways they could spin a company up off of this. And the Institute was interested in doing that. And that's when, I think in early 2018, when he and I were introduced through a mutual friend. And the rest is history from there.
I find it interesting because you did mention hair follicles at a bit of a white space, dermal papilla in particular. Do you have an opinion on the sort of the human scalp skin organ culture model or the hair follicle organ culture model as being potentially more applicable to understanding human disease and human conditions than just solving hair loss, or do you have any perspective on that?
I do. We actually talk about that when we're dreaming about long-term, decade from now, how could the work that we're doing extend beyond just hair? And we really think it does. And this isn't, obviously we're biased. We think that, our breakthroughs in technology development are going to be important for a lot of space, but we have strong networks across the scientific community, almost none of whom are involved in hair. And they're extremely excited about the broader, what I'll call tissue engineering capabilities that are going to be resolved and discovered by a process that can master the formation of hair. And so I think the one thing that folks really do understand is how complex the hair follicle is. And the complexity of this little tiny mini organ is equivalent in many ways to our larger organs. And so if you can resolve ways in which you can engineer the cells and then take those as building blocks to engineer a functional, durable little organ, you can take those principles, you can take those tools and techniques and protocols and start to apply them to larger organs of similar complexity, but larger in, physical size. Think of kidneys. We have some colleagues working on kidneys. There's a number of companies trying to regenerate brain tissue to solve Parkinson's and others. There is, I think, a broader capability that is going to be informed by some of the principles, scientific principles and engineering techniques that we discover here at Stemson. So that's one way to think about it. And time will tell whether we're right or not. But we'd like to think that that's going to be one of the benefits stemming out of our company.
I love that you guys are thinking about that, thinking about it like that. And it's a little, it's validating of, I think, the way we talk about it a lot as well, because it's, the hair follicle is so accessible and it's so scalable in terms of being able to test a lot on it and easily get sort of, samples of it rather than, for example, flipping off a part of somebody's kidney. It's, or a little punch. Here you go. Here's my hair follicle. You can test on it.
Yeah. I'll say one thing. If we as a human race cannot figure out how to regenerate hair and create this durable structure, why would we think we can do something of a larger organ that is inside the body that's really difficult to access and may even be more complex? It's kind of like the hair follicle is the first logical step to demonstrating a true regeneration of a fully functional organ, that we should focus on solving that first as a call it a baby step towards thinking then a broader body regeneration.
Yep, totally agree. Okay, so moving sort of back to Stemson and a little bit away from the preclinical model, and I've heard you talk about this a bit on other podcasts, but since the founding of Stemson in 2019, what would you describe as sort of been the biggest challenge the company's had to deal with? This can be like there's definitely scientific hurdles with hair cloning, but then also like a lot of hair loss companies sort of get lost in the annals of history for non-scientific reasons, IP getting held up, for example, or whatever. So would love to hear your thoughts on that.
I think I'll point to three. First one is, interestingly, finding talent, finding people that have not just some expertise in hair and the field, but also have some product development and industry expertise, which is a different skill set than you might find in an academic lab in general. So we've almost exclusively had to hire folks who don't come from directly in the hair field. They just don't really exist in industry. And there's some in academia that we have sort of pulled out of academic labs. But for the most case, we've had to take people from similar technical challenges, but for a different application. So our chief scientific officer, Kevin, for example, is an expert, a global expert in differentiating pluripotent stem cells, but has never done it for hair follicle cells. He's done it for pancreatic cells and for other immune T cell related products in cancer. But the techniques and principles of differentiation are the same regardless of your target cell type. We've had to bring bioengineers in that have some skin expertise, like our chief development officer, Megan, but have never applied it specifically to hair. So we had to bring some talent together, and then they had to kind of learn the principles and basics of hair and the biology behind hair while they were here. So same is true for me, quite frankly. I don't have a background in hair follicle biology. My background is in genetics and how we study the genome and its effects on diseases. So that's one. Just finding the talent has been a challenge, and I think we've been successful up to this point, but it takes time versus some ready cancer expert company that has developed cancer drugs that can start on day one. So that's number one. Number two, on the scientific front, Andrew, you and I were talking about this earlier, the belief in some preclinical in vivo animal models that could actually reveal to us useful information about how a product would behave in a human being has been really difficult. And we bounced around from early days in a few different animal models, from mice to pigs. We've finally settled on, after quite some time, a human skin xenograph model in a mouse that we've spoken to the FDA about this and many other experts, where everybody's basically like, that's the best we're going to get, is to recreate a human skin environment that has the anatomical structure and thickness that you cannot get from mouse skin, for example. It's just too thin. We physically can't fit our product into the mouse skin. You need a thicker skin pad, and so that was another big challenge that took us quite some time to get through, and wish we had been more confident on exactly what that was from day one of the company. But we're there now, and really excited about the path forward. And then so that was number two. And then I think the third area is just in the investment funding side, right? I mean, funding a therapeutics development company is about the longest and among the higher risk investments you're going to make. And the number of people in our field, myself and our board members and others have a deep network of biopharma, biotech, venture capital funds and investors, and almost none of them have done any real work or any knowledge in hair therapeutics. So back to this challenge on getting talent hired into the company, it's similar on the investment side, where everybody understands and gets the problem of hair loss, but when they look at it to evaluate from an investment perspective, the risks associated with it, how can we be confident in the technologies? It takes some time, right? And, this lack of animal models makes them nervous. They're used to cancer, where the animal models have been settled and developed for decades, and you can get readouts you're confident in that have some history of translating to humans. And so that's been a struggle to find the folks who are willing to kind of dive in and take a risk. But the cool thing is, we're sort of leading the space in this big, broad, exciting market opportunity. Folks believe in the market opportunity and the payoff from a patient perspective, the payoff from a financial perspective. So there's enough willing to take the risks there. We just have to kind of take some time to find them.
Yep, absolutely. No, I do think it was a really interesting point you made when we were talking about the preclinical models before, is that, like all of these institutions, the FDA might need to evolve, and, animal studies make a lot more sense for safety than they do, at least for efficacy, particularly in the hair loss space. And the fact that there's been thousands of hair loss companies that have, fully regrown hair in a mouse, and, we still don't have a treatment. So I got you. So touching a bit on your mouse human skin xenograft model, I believe you made the announcement in February, I think, that you guys had successfully transplanted the hairs onto the mouse xenograph model. Can you just talk about, because you guys have had done similar things in the past, so can you talk about sort of what was sort of the breakthrough of this study, and generally like what the results were you saw in terms of hair quality and all of that stuff?
Yeah, absolutely. So let me first put the results we announced last month into perspective, where the results we achieved were using the actual product, three-dimensional physical product construct that we plan to use in humans. And I'll get to that in just a moment. Whereas previous results were taking components of the product, especially the cells, and sort of seeding them into this human skin as it was sort of healing and forming the skin on the back of the mouse, which doesn't represent adult skin, right? You're not transplanting into skin that's in like a healing phase, right? So creating human skin on the back of a mouse that can survive and be durable for months is a complex process. And so we've had to do some innovation there. So what we started with was pre-seeding those human skin pads. At the same time, we transplanted the skin itself onto the back of a mouse with also our folliculogenic engineered cells. And so that just tested whether the cells that we engineered were competent to grow hair. However, that's not what you do in a product or a clinical setting. So instead, of course, what the actual product will be these bioprinted engineered follicular units. And so that bioprinted unit that looks like a little hair, it's literally biomimetic. These things are tiny, a little less than the size of a normal human hair. And we have to combine the cells with some biomaterials to enable handling and transplantation. And the cells need to survive a brief inflammatory period when you create a minor wound to poke the skin. And then they need to engraft, and then they need a number of weeks, potentially up to 2 months, to actually form the hair shaft to come up and out of the skin. And so what we did last month was we demonstrated that the product concept, the actual product we would take to market of these three-dimensional constructs, which is think of it as a ball of cells with this little scaffold structure that helps enable directional growth, transplanting rows of those units into a human skin pad that has been fully healed, has been settled for a couple of months so that it can more closely mimic normal adult scalp skin, and putting those units in and then demonstrating over time that they generate healthy, normal growth human hair follicles with hair shafts and have all the right appendages such as a sebaceous gland and these things that you need for healthy, durable hair. So that's the difference. I hope that makes sense.
I didn't quite get that from reading the press releases, but so I think a lot of people would be very excited to sort of hear that. That's the update. Okay, so now that you've successfully transplanted the go-to-market product in the the fully healed human skin on the mouse's back, what do you foresee being the biggest hurdles or challenges in between this point and I guess both sort of getting to a phase 1 clinical trial and then also just like on the market, fully on the market?
Yeah, the path is now pretty clear what we need to do. What we're really focused on right now is finishing the final tweaking of the product and the process to make the cells such that we can move it into official IND enabling studies, which are sort of a more formal governed approach that the FDA requires for a third party to do those tests in an animal model. And so what we know is remaining to do is, because we are designing an autologous product, meaning we will use each individual patient's own cells to create the product, we now need to demonstrate that our process can be consistent from patient to patient to patient. People may be familiar with the concept of high variability from patient to patient. This is common across the industry, across pharmaceutical company products. It's common, for example, from mouse to mouse in mouse studies. And so humans are no different. So we need a process that is robust enough where showing it working in just 2 human cell lines is not quite enough. We need to show it across dozens, and we may need to make some tweaks to the process to make it more robust to deal with some of the patient to patient variability. And so that's really the most important aspect from here to being confident that we have a cell differentiation process from IPSC to hair follicle cells that can handle any variability that may come our way from patient to patient, and that it ends with the right cell identity and cell purity at the end. So there's some tweaking to how that needs to be done across patient lines, and that's the core. And then hopefully we get done with that. I mean, we're racing as fast as we can to be done with that, so we could try to get into IND enabling studies ASAP.
Gotcha. Okay. Fascinating. Awesome. And then, so, a big part of the complexity I would say that Stemson's overcome, which has been so impressive, is the fact that you guys are really you're growing the multiple cell types that all go into a hair follicle, right? And to create this sort of final hair follicle. And as our community knows, right, the hair follicle is not just one cell group. It's a bunch of different cell groups talking to each other. I'm curious, at any point in Stemson's development, because there are other companies who have pursued a slightly different route. Did you guys ever consider sort of trying, growing, for example, the neo dermapapilla, where you're only using dermapapilla cells to then regenerate the entire follicle? Did you ever have a path that went through using one cell type to clone these hair follicles?
Yeah, we have tried this. And trust me, we would love to only have one cell type to deal with. This would make the product less expensive, for example. It would make the whole process less complex. And so far, our own studies in our own hands have shown what the field seems to have been saying for a long time now is you need at least two cell types. And so right now we are, what has worked in our own studies has not been a dermal cell type alone, has not been an epidermal cell type alone. You need one of each. You need an epidermal cell type and a dermal cell type. And now what we are seeing is that that epidermal cell type and that dermal cell type are progenitor in nature. They are not terminally differentiated adult-like cells. They're actually, because hair follicles and the structure of the hair follicle tissue forms only once in our life cycle, which is during our fetal development in utero, you actually do want to target, and this is the genius of the beauty of IPSC, is they're making fetal-like cells, if you will. You want to target sort of that early dermal progenitor and an epidermal progenitor. And if put into the right structure and the right environment in the skin, they kind of know what their job is. And they will then further develop and differentiate into the various more terminal states of the structure and layer of the follicle. So the dermal papilla compartment, the outer root sheath, the inner root sheath, the terminal keratinocytes that form the hair shaft come from those two progenitor cell types working together.
Yeah, I figured it. Like I figured you guys would have gone the more simple route if it was feasible.
Yeah, we would love to. I mean, even the whole industry is not even perfected how to manage one cell type. Like it's not simple because cells don't always like doing what you tell them to do. But we think that two cells are going to be required, and we would love to be surprised and find a way to do it with one cell. But right now we're pretty clear on a path forward with two cells. We know that works.
Got you. And so now to kind of go on to the induced pluripotence, and I know you guys were initially experimenting with blood cells, like using the existing phlebotomy network, easy to transport and all of that. I think Kevin, your CSO, in a recent interview said that it might be difficult because you guys had found blood cells might have sort of an epigenetic memory, which could then make the redifferentiation difficult. Correct me if I'm wrong on that interpretation. But just curious to hear your thoughts a little bit on that struggle and any plans you may have to leverage a different cell type's existing network or anything like that.
Yeah, I love this question, Andrew, because this is still a very nascent element of IPSCs that the whole field are figuring out. And there's been some seminal papers coming out from a few different labs just in the last 2 years about the potential that the starting cell type you use could possibly be preserving some level of memory of what it was after you reprogram into IPSC, and that memory could cause some malfunctions in the cell after you differentiate it to a different cell type. And so a couple the early discoveries on this in the field were really for T cells, because T cell engineering for fighting cancer has been the forefront of all of cell therapy. And so there's a couple papers that say if you start with a T cell and you reprogram it into IPSC and then you redifferentiate it into a T cell, it appears to have a better function than if you started with, say, a blood cell or a fibroblast. And so in our own hands, we don't know yet. Quite frankly, there are really just two options. We're going to be having a bit of a bake-off to see what is the right starting cell type. Blood is one, and the reason we've communicated that publicly is just because of the ease of capturing blood, preserving it, cleaning it, transporting it. I mean, we do it every day, everywhere across the world. And there's a great supply chain to do that. And there's also very good protocols to create IPSCs from PBMCs captured out of blood. However, we've not locked in this decision, and we actually are quite curious to see if fibroblasts, knowing that many of the hair, especially the dermal compartment of the hair follicle, is a special fibroblast type cell, that we do have some IPS lines in that were derived from fibroblasts. And there's very robust protocols to take fibroblasts as your starting cell, reprogram them into IPSC, and then we can differentiate from there. So, we're going to just basically do some in-house testing to see if it matters. If it doesn't matter, we'll probably go with blood because that's the easy one to do. But if there's actually a difference in performance, both with the in vitro culturing process as well as the in vivo outcomes that we test, if it proves to be superior using fibroblasts or some other cell type, we would settle on that. So that's an ongoing, as Kevin signaled, sort of open area we're trying to lock in sooner rather than later. And we have, cell lines from both starting cell types to help us answer that question.
Got you. And then have you guys thought, just in terms of like the fibroblasts, do you, maybe you've seen something or maybe you have a hypothesis about this. Do you think there would be a difference in terms of inducing pluripotence, for example, from like occipital fibroblasts versus frontal fibroblasts? Like, do you think if you like the hair clone follicles might be more DHT resistant, androgen resistant if you did induce pluripotence from an occipital fibroblast? Or maybe there's just no data to support this.
Yeah. So just to be clear with you and your audience, there is no data to support this right now. So like the answer really is we don't know, but I'll tell you what we hypothesize. And again, we'll probably do this testing ourselves at some point in the near future, is if the reprogramming from a starting cell type to induce pluripotent stem cells is truly effective and efficient, it should not matter where you capture that fibroblast from. And as far as I'm aware, the androgen sensitivity of the follicles in the front of the head and the insensitivity in the back of the head, we don't really understand the biological mechanisms behind that. And so no one has sort of clarified or revealed from a genomics perspective or other cell biology perspectives, or tissue biology for that matter, maybe it's beyond just the cellular level, what is resistant versus non-resistant? How do you define that? And knowing that we are just trying to get to a good, highly pure IPSC that has roughly erasure of its previous identity and functionality, it shouldn't really matter. And so we'll just have to use our own empirical data to test that. Perhaps there will be some data coming out from other labs that can inform us on that, which would be great. We'd love to better understand. We don't want to have to do all the research ourselves to know what is insensitive versus sensitive. Like we'd love for others to tell us what that mechanism looks like, and we'll try to engineer towards it.
It's funny. We're all really definitely operating on the edge of the unknown. And so often the answer will be like, you tell us. All right. And then, okay, before I shift it over to Bax, who I know has a bunch of questions for you. The last thing I just wanted to really understand about Stemson, and I think I know the answer, but like it would be really helpful to hear it from you. So Kevin referenced that you guys have outsourced the reprogramming of these iPSCs. And so I want to get, because that seems to be like pretty well established protocol, like across outside of hair follicles as well. So can you just touch on what is the proprietary part of the process for Stemson? Is it the very initial stages of redifferentiation, or I'll let you talk about it? Yeah,
you're right. We are not innovating on going from starting cell type to iPSC. There's tens of billions of dollars across the industry working on that step. We want them to figure that out. We want it to become commoditized and standardized and just let the best do it for us. What is proprietary and protected is pretty much every step, starting with iPSC down to the finish line of the product. So how you differentiate from iPSC into the dermal progenitor cell, patent protected. That was actually the core patent behind Alexi's discovery at his Sanford Burnham lab. We spun that out of Sanford Burnham to help found the company based on that. Stemson itself, after forming the company, created the protocol to differentiate from iPSC to the epidermal progenitor. So that arrives at our two cell types. And then we have that last step where we have to form these three-dimensional constructs that are bioprinted. That is also a patent-protected step in the process. How the cells are combined, what they're combined in, basically the process behind doing that to arrive at the final product. And so that's the core of our proprietary protection, not including a number of patents we just took over from the Aderans deal. That's a separate thing.
Very cool. All right, Bax, over to you.
These are more questions from our community. So we kind of vetted some of those. I guess based off like the demand for the answer, but I think the biggest one people are most curious about is your roadmap. And so maybe if you could just get in like both the near term, like what are the goals in the near term, and then also I think people are really curious about just like sort of like the output. So because we see these press releases with the mice regrowing the hair on the xenograft. Like, and obviously you're going to be testing new cell types. I'm sure there are other variables that you'll be changing too. Like how many of these mouse studies can we see? Like, like when should we expect to see them? That sort of stuff.
Yeah, absolutely. I mean, everybody wants to know when is this going to happen? When's that going to happen? We do too. It's, in novel science, it's hard to predict timelines. You can't control per se when you're going to achieve the success. What we can control is how many activity cycles we can rip through, in a period of time. And I can't predict whether we'll have success on the 5th try or the 25th try, right? That's the hard part about novel science. So bear with us as we think about roadmaps. I'll tell you kind of the core goals. The number one goal for this company, for our core foundational program, which is this iPS-derived product to generate de novo hair follicles, is to get to phase 1 clinical trial outcomes. So use that as an anchor in the future, that if we demonstrate proof of concept, even at low efficiency rates, right? Like if we test it on 10 humans and it has a good outcome in 2 or 3, like that actually shows this is possible. Obviously, we want it to be better than that, but I'm just trying to set some expectations that demonstrating successful proof of outcome in human beings is what solidifies this company and our success and existence into the future. So we're really focused on that. How do we move backwards from where we are today, from where we want to be at that point in the future to where we are today? And so what we're really focused on is lock in those final few parameters to do patient to patient, cell line to cell line variability in our process so that it can be robust to handle it. And then as soon as we can, hopefully next year, we'll be initiating those IND-enabling studies, which puts us on the path to submit a clinical trial application. Those studies can run anywhere from 6 to 9 months as per required from FDA, or, we actually might consider going into a different country for financial and timeline reasons. Others allow for slightly shorter timelines and offer financial incentives to go into their country. But use that as the anchor, 6 to 9 months for safety and efficacy studies that will be conducted in these mouse models that we've created. And then you have to validate a GMP grade manufacturing process to make every component of the product and make the final product. And that takes some time too. That's actually a pretty heavy investment to get something up and running and validated from a manufacturing perspective, because all that manufacturing data then goes into your clinical trial application together with those IND-enabling animal study data. And that those two main components are most of what the trial application is. And so we'd love to be into trial, as soon as possible, probably a couple of years away, just to try to get through all those things that I just mentioned. And the trial itself, we'll see what the regulators, agree to. But, we think we're going to be targeting both a safety and an efficacy endpoint in this phase 1 trial. So we refer to that as a phase 1/2 trial. So where you're taking elements of a phase 2, and this is mostly common in cell therapies, because you're putting a functional cell into the body. We may as well measure its function, beyond just safety. So we'll do this phase 1/2. Primary endpoint being safety. Big safety issue you got to show is that these are not genomically unstable cells that could cause tumors. That's the biggest safety concern. Tumorigenicity is what they call it. That'll be the main thing we're looking for on the safety side. Then secondary endpoint on efficacy is actually quite straightforward. Did it grow hair or not? And a lot of different ways to think about that. But so that's really kind of the core roadmap. Obviously, we now have another program that we're bringing into the company through this Aderans deal. We can talk about that separately, but we are all systems go. Even with the new Aderans program coming in, our plan is still all systems go on this iPS-derived program to move it forward together in parallel with this other program from Aderans.
Yeah. And would love to hop into the Aderans in just one sec, but just so on the timeline here, to get to the IND enabling studies, there are a certain amount of mouse xenograft studies still that we need to run, right? How many of those, and if you can't share this, totally cool, but like how many of those do you think are, or maybe not, maybe that's not a fair question, but I guess what is the rate of output of those studies, I guess, if you can share that. So like are we looking at like 1 a month, 3 a month, 5 a month, 1 every 3 months?
We have the capacity to do several per month, but here's the important rate limiting factor. A single in-life study from the moment you transplant to the moment then you sacrifice the mouse to study what just happened inside the mouse is typically 10 to 12 weeks. So this is just the speed at which the biology goes, and we can't really control for that. Kind of like you can't take 2 women and make a baby in 4.5 months. So, so the key for us is, there's a certain period of time to create the product. And then once the product is ready to go, then you have to transplant it. Then you have to wait a couple of months for the in vivo outcomes. And yes, we do some time course studies along the way to see, kind of what's happening at various points along the life of the mouse. But the endpoint is really 10 to 12 weeks out. So that's a long cycle, right, is my point. And so the key for us is how many of those can we run in parallel? What's the resource level we have to take? If we have 10 different ideas we want to test, can we do all 10 of those in parallel? No, not today. We don't have a big enough team to do all that. So like how many can we fit in with the resource levels we have? And that's where we're just working through that as much as we can. And, if you get positive outcomes, that advances things rather rapidly. If you get negative outcomes, you got to kind of go back and say, okay, what did we learn from that negative outcome? What do we go back and try next time? And as anybody who's done a lot of scientific research, product development, you actually get more failures than successes. They got to have a sturdy, sharp resolve in science here and product development to get through that. And that's just kind of part of the journey.
Yeah, no, that's very helpful. So we'll look out, 3 months from now for the next one.
Yeah, sorry, just a real quick question, because I know we also have a bit of a crypto audience, and you mentioned you were considering other countries. Which countries were you thinking of in terms of being sort of friendly from a regulatory perspective?
Yeah, we have a couple in mind. First, I'll just state that we are looking to go to a country that has a robust and established regulatory regime, especially in cell therapy. So just want to be clear, we're not trying to go off the map to some, somewhere that these kinds of trials have never been run before. That's not what we're going to do. We're going to go look at places like Australia, like Singapore, like UK, and Canada as examples of a few other places. And there's different reasons why we would look at those. Many of the factors have to do with, is there any kind of established infrastructure for cell therapy manufacturing? Making cells is entirely different than any other pharmaceutical drug modality. And so having no infrastructure, no facilities, no human resources in the area that knows anything about cell manufacturing is probably going to make it extremely difficult to consider that country. But the ones I just listed off are pretty robust in this space. And some of them, like Australia, have very robust financial incentives and rebate programs to pay for your manufacturing and clinical trial. That's why we would, consider a place like that.
No, it makes perfect sense. Really smart. And then, so moving on to community questions from Herbert and Donna, who are two of our biggest stem cell experts. They really want to know how we can support the Stemson mission. So, anything about the current fundraising status, helping recruit patients, tissue, talent, what type of talent you're looking for if you're looking for that right now. Just any way we can help today.
Yeah, really appreciate this. And, it really does take a community and a village. We prefer not to have to tackle all the challenges ourselves. So we really appreciate the help from you guys and your community. I would say we are in fundraising mode this year. So we want to, especially with the new Aderans deal and bringing in a new program, we want to make sure we have the resource level to carry 2 programs forward. What I'll just say about that, though, is the amount of funding we're pursuing will be large, right? Tens of millions. And so if anybody has, any particular ideas or contacts about funding sizes of that amounts, we're always open to that. But I just want to be clear that, offering to get 50 grand here or 100 grand there is not going to be, not trying to be disparaging, but just won't really be helpful to get to our goal unless there's some massively scalable way to do that, right? So that's one I'll point out there. The other one would be talent. I mentioned that as a challenge. And so when we secure this new funding, at some point down the road, we will absolutely be going on a hiring phase. And so I think not so much right at this moment, but maybe, 4, 5, 6 months from now, we might need some help in bringing some talent to the space. And that talent will be in a couple of areas. One, folks that have a robust background in differentiating pluripotent stem cells would be one thing that's very helpful. Folks that have a strong background in hair follicle biology that can translate into product development. That's extraordinarily rare, but just bolstering the hair follicle biology capabilities and expertise inside the company would be really helpful. And then I would say the other big area will be, we're going to be moving into a manufacturing phase. So people with expertise and experience in cell manufacturing would be another area, because that's part of what we hope this next round of funding will enable, is for us to move out of just development, research and development, and into road mapping out what does it look like to actually have a GMP compliant cell therapy manufacturing capability inside the company. So those would probably be the most immediate areas that I'll point out.
One question also from Herbert and Donna on the GMP manufacturing. Will that be for the autologous treatment or for the allogenic treatment? Because there have been, that's something that has been discussed quite a bit.
It would be the autologous treatment. And yes, we do have plans in the future to spin up an allogeneic IPSC program that will start all the way back at the beginning of the research phase, just because you have to figure out how to edit the genome of the cells to make them immune evasive, and then ensure that you do the testing such that the cells are safe, which is the big concern for doing gene editing, but then are functional to form a hair follicle. And then it will require its own separate clinical trial path from the autologous solution because of those safety concerns. With gene editing, it's considered by any regulatory body to be a separate product from an autologous product, and it requires its own testing. So this, in the near term, it will just be the autologous manufacturing process we'll pursue.
And so then just two more questions. So with the Aderans deal, could you just get more into, obviously, you're doing a lot of stuff with the IPSC work already, like getting this to clinical trials. How does that play into the grand vision?
Yeah, we're really excited about this deal. Spent a lot of time working with the Aderans team to figure out if this is something we should do. Let me just state first and foremost that Stemson's mission is really all about bringing cell therapy solutions into the field of hair loss. It's not, it's not a single technical way of doing cell therapy that we're wed to. Like, it doesn't have to be IPSC. We're just any way in which we can leverage a cell replacement tissue kind of regeneration approach makes sense from our mission and our capabilities. And so we had actually been thinking about several different product concepts using live cells as the medicine to then address different forms of hair loss. So not just androgenetic alopecia, but alopecia areata. You could imagine an allogeneic immune evasive cell being potentially successful at doing that. Or in scarring tissue, there's a history of hair transplants into scar tissue that have been successful. What if we could make our solution capable for that? We also had thought about a simpler delivery. What if we could just take the cells that we make and just inject them into the scalp somewhere? Could they have an effect? And that's exactly what Aderans was doing. They didn't use iPS cells to do it. They used adult excised hair follicle tissue as their starting material. and it was autologous. And through a culturing process, they created a cell product that then, through a simple injection into target areas of active hair loss, attempting to rejuvenate or repopulate those shrinking hair follicles with functional hair follicle cells that had been degenerating away. And so, knowing that, Aderans decided that this program, for reasons we won't get into, kind of got shut down for a long time. And they were looking for, when I met them in 2022, they were looking at possibilities of resurrecting that program because they had spent so much time and effort getting it all the way through phase 2 clinical trials. But they're not a therapeutics development company. They're the largest wig manufacturer in the world. They own Bosley clinics. They do a lot of hair care services and products, but not therapeutics. And they had decided strategically, like, we're not going to be the ones who kind of take this development forward. But Stemson looks to be the leader in this space, and that opened up the conversation for us to think about whether we should do a deal like this and should we bring this program in. And we see a lot of different synergies and complementarity between our IPS approach and then this adult hair follicle cell approach that really is more specific to targeting early stage androgenetic alopecia in men and women to kind of repopulate follicles that are in an active shrinking state. So not like late stage, hair loss where your head is pretty much fully bald. You'll need our IPS derived product for that condition. But think of it as an approach that tries to stop hair loss, from the beginning. That's the Aderans approach versus Stemson's IPS program is kind of like, don't worry about what's happening with your existing follicles. We'll just replace them for you.
No, well, if you need a test candidate for the Aderans program, I think I could be I think I could be right up your alley.
Yeah, there's a big need there for sure. And I think better solutions than, say, what Rogaine or PRP is able to do today. It's just not satisfactory enough. And this Aderans product would have a kind of one and done, just a single injection of the cells, and you get a long lasting, durable effect. Can't perfectly predict whether it'll last forever, but they certainly have some interesting clinical data that shows that it can last a long time.
Will you be bringing that to market anytime soon?
That's the idea. Yeah, we one of the interesting things for us is we captured a program that basically got at the end or towards the end of phase 2 trials. We're going to have to make a strategic decision when we get that technology in our own hands around, should we repeat a phase 2 and then go into a phase 3, or should we just go straight into a phase 3? It's one or the other. But yes, we will be planning to take that product forward towards commercialization.
Awesome. Well, yeah, I look forward.
Will that decision kind of be based on the homing of the cells to the appropriate and actually like the durability of their integration, or like, is that what you will be sort of looking at with the Aderans IP in terms of, do we take this straight to a phase 3 or not?
Yeah, it's all going to boil down to confidence level and data, both the human data from the phase 2 trials as well as some animal related data that they had produced, as well as we might just do some rather quick testing in our own hands to give us the confidence of the efficacy of the product. The product is already proved to be exquisitely safe, which is wonderful. So, we're not concerned about that. It's all about optimizing for effect in people. And so if we can get enough confidence that the effect looks pretty robust, we might go straight to a phase 3 trial. It's about like phasing your level of investment, right? Obviously, a phase 2 trial is less expensive, a little less risky than a phase 3 trial. Do we want to kind of get more confidence before going big into the phase 3, or do we want to just go straight into it? So that's a little bit the analysis we have to do.
Well, this is our last community question, and I think it plays well here, is from Cat Belly Rubs. So especially now with the Aderans partnership, and then shortly after the IPSC program, do you have like a network of transplant surgeons to administer these treatments? Like, let's say I want the Aderans treatment. Where do I go to get it? What's the distribution here?
Yeah, you're talking at commercial phase. What would that distribution be?
Yeah. So once for instance, once Aderans, and then later once the IPSC treatment is ready.
Yeah. So for the Aderans product, we expect it to be a wide network of dermatologists mostly. It could be other cosmetic doctors. You don't have to be a dermatologist. It's essentially just an injection intradermally into the skin, which, there's so many products out there that do that. And this is practiced. You could probably go to a couple of square blocks in New York and find, 10 doctors that do it right there. So we expect the Aderans product to be a rather simple injection through a syringe, needle-based product that can be done by so many different doctors, mostly dermatologists. So we do expect that to be widely available. With the other product concept, that we founded Stemson on, the IPSC derived, that's a little more complex and likely needs more training and expertise that you'd find typically with people who have done hair transplant surgeries. So we would likely put that into the hands, first and foremost, of the best hair transplant surgeons, and you need some tools, perhaps some robotics to get scale. If you want to transplant 10,000 units into the head, for example, you don't want to do that by manual hand. So we'd probably start by looking for the best hair transplant surgeons, and then look to expand from there. Obviously we'd want to make it more and more available over time in more areas as demand grows, and we add those capabilities. But so a slightly different approach to the two products and in who we would enable to deliver it early.
Were there any discussions of potentially tapping into the Bosley network for your autologous iPSC treatment?
Yeah, sure. I mean, nothing in the agreement related to that whatsoever, but we would for sure look at Bosley as a leader in the space, a leader in hair transplantation capabilities, and we would look at them as one of the high potential early adopters to deliver that kind of product out into the market for sure.
And then I guess how do you think this will impact sort of the autologous iPSC progress, if you will? Do you guys have the bandwidth? Do you need to add capacity to kind of bring these two products? I mean, I guess it's what the fundraise is probably.
No, we need to add capacity. I mean, we're not resourced currently to do two parallel programs of this nature. So we'll definitely need to add capacity. But we do believe that having both of these programs under one roof offers a tremendous amount of synergy where you can develop both programs together faster than you could separately, because there's so much overlap between them, and so much sharing of knowledge capabilities between the two programs. So that was one strategic reason why it made a lot of sense. But we're not going to scale up the needed resources for both programs until we ensure we have the right funding in place, and that's part of the game with startups.
Gotcha. Okay, I guess I just had one question that if I don't ask it, Indana would probably kill me. But so the question for you is sort of everything works out perfectly. This is Geoff's dream world. Can you take us through sort of what either the autologous or the allogenic final product ultimately looks like? From a patient perspective, you go into the transplant clinic. How long does it take? What does it cost? And then also I guess this is just one other thing is, because your sort of iPSC follicles are actually thinner than a normal follicle, will you guys be able to increase the density of hair on the scalp as opposed to a normal transplant?
Let me answer the last question first around density, and then I'll get to, what does the patient's experience look like? We believe the answer is yes. Through the dose of cells, the number of cells that we put into the individual engineered units, especially the number of dermal cells, has been shown through studies to have a direct effect on the thickness and robustness of the hair shaft itself. So in other words, typically larger dermal papilla compartments mean bigger, sort of more robust hair shafts. So you could control that parameter theoretically. Obviously, we need to show that it can be done in practice, right? So that's that piece, and that is an engineering aspect of the product that we do have our eyes on to optimize down the road. Back to kind of the patient's experience, what we really are aiming for over that long term is that the patient has an opportunity, if they prefer to use we want to give them a choice. Do they want to use their own cells to generate the solution, which is going to be a bit more expensive, frankly? I think people understand that. Or are you okay using someone else's cells to generate this product? The sacrifice you'd make there would be that you can't always perfectly predict how someone else's cell line is going to operate in your own body. Over time, we would hope to demonstrate high confidence in that. But you gain by getting a less expensive product, and you can have it delivered to you sooner, right? So we want to give them the choice. We think there's actually going to be a market and a preference for one or the other. If you really ask somebody who has the financial means, would you prefer having your own cells or someone else's? I think a lot of people would choose to have their own cells. We want to give them the choice. And so a little longer duration for an autologous product because you have to capture someone's biological sample and put it through the process. So that's likely going to be, probably something in the range of 6 to 8 weeks before, you have the product ready to be delivered to them. Versus in an allogeneic setting, you can do it probably within just a couple of weeks to place the order, get the product made and delivered. But that, we want to offer this in the price range of today's hair transplant procedures in the long run. Any technology sometimes takes a little bit of cost curve, scaling over time probably won't be too much different than that. Something starts off a little bit on the expensive side and then bring it down as we scale and we optimize and improve the technologies. But that's the goal, is to get it into that range of hair transplant surgeries for something of that huge clinical value. This would be a massive clinical breakthrough in order to do this. And so we think that hair transplant procedures as they exist today will basically phase out over time. And instead you would just get the Stemson treatment. And, we're excited about that opportunity alone, because I think it's a couple of 100,000 people in the United States every year get a hair transplant surgery, much larger from a global perspective. But I think once you demonstrate the breakthrough clinical value, you'll get even more people interested who previously wouldn't do a hair transplant surgery. They just didn't want to go through that process. But with an ST1 product, which is what we refer to as our IPS program, the benefit of like a rather rapid delivery of these units, and you can avoid any extraction out of the back of the head, could be bringing a new set of people into that market.
We can do that. Well, whoa.
Yeah. I just knocked you out of your seat, Andrew.
A few moments later.
One more question to let. So I think you had mentioned that the Stemson hair germs are 1/3 of the width of the terminal hair follicle. So instead of the actual diameter of the hair follicle, I think the best hair transplant surgeons can get something like 80 follicles per square centimeter or something. Have you guys thought about do you think it's possible Stemson could get up to 120, which just because of the germs being 1/3 of the size, actually getting back to the full healthy head of hair?
I think it's possible. I don't want to underestimate the challenge of achieving that level of density. And the only reason I say that is because you have to use a needle to insert these, and there's always a risk that the needle could damage the existing other follicles around the area. But I absolutely do think it's possible. We may need to miniaturize even a bit more from where we are today. And I would say, early commercialization, we probably wouldn't target, that level of density. But over time, in the spirit of improving, the outcomes for patients, we would look to improve the density. I think if we can just give, at least today's standard for hair transplants, and we can match that to start with, but cover, much larger parts of the head, that'd be an outstanding starting point. And it's just kind of hard to tell, Andrew, whether we'll be able to get there, but I do think it's certainly not out of the realm of possibility.
Cool. Amazing to hear. Those are all the questions I had. Bax, do you have any?
No. I'm all out. Thanks so much for the time, Geoff. That was great.